Start now.
Okay, sir. Good morning, students. Welcome back to another session. This is Vani from TSRJC, Wyra. We are going to the class. Before that, I would like to appreciate Pooja, Nirosha, Durga from TSRJC Hasanpatti, Asma, CH Narmada, Grishma Sri from TSRJC Wyra, and one more Harshini from TSRJC Burgampad. Very good, students. Well done. Yeah. This is last session, that is sexual reproduction in flowering plants. Before class, we have discussed in pre-fertilization structures and events. So now today we discuss about the topic pollination and post-fertilization structures and events. So what is pollination? Do you know? Already we have learned before classes what is pollination and importance of pollination. Yeah, here pollination. Pollination is transfer of pollen grains straight from the anther to stigma of pistil is known as pollination. Pollination means transfer of pollen grains from anther to stigma of the same flower or another flower. This is known as pollination. In this process of pollination of fertilization was first discovered by Camerarius. Okay, students, this pollination in definition, transfer of pollen grains from anther to stigma of same flower or another flower. Next, what is the importance of pollination? The importance of pollination, first one, it is a essential step towards to sexual reproduction, and it leads to stimulation, germination of pollen grains to produce male gametes. Male gametes, and it helps to bring male and female gametes. It is close to fertilization. The pollen grains, it reaches to anther to stigma after the process is completion of fertilization process. This is importance for pollination. We will go with next slide. See here, this is pollination. Pollination is divided into the two types, self-pollination and second one, cross-pollination. So what is self-pollination? See here, self-pollination. When the pollen grains are transferred from the anther to stigma. See here, this is male gamete, this is stigma, female reproductive organ. When the pollen grains are transferred from anther to stigma of the same flower or another flower on the same mother plant, it is called self-pollination. This same flower, this is another flower of self-pollination. This self-pollination againly divided in two types, orthogamy and zytonogamy. So what is zytonogamy? When the pollen grains are transferred from the anther to stigma of the same flower, it is called orthogamy. It is possible only bisexual flowers. So bisexual examples for Pia, and hibiscus. And one more example, solanum. This is autogamy. And next one, self-pollination, zytonogamy. So what is zytonogamy? See here, when the pollen grains of flowers are transferred to the stigma, another flower on the same mother plant. It is called zytonogamy. See here, this is one flower, this is another flower. The anther, the pollen grains transferred to stigma of another flower. This pollination is known as zytonogamy. Example, cucurbita. And one more example, maize. Okay, students, this is self-pollination. Self-pollination is two types, autogamy and zytonogamy. And next one, cross-pollination. What is cross-pollination? See here, this is cross-pollination. When the pollen grains of the flower of one of the plant and the transfer to the stigma of the flower on the other plant of the same or different species. It is called cross pollination. So here this is one of the flower, this is another plant of the flower. In this cross pollination, it is occurred, transfer the pollen grains, stigma to anther to stigma of the flower, other plant of the same or different species. It is called cross pollination. So againly the cross pollination also divided in two types. That is first one xenogamy and second one hybridization. So what is xenogamy? See here, when cross-pollination takes place between two plants of the same species, it is called xenogamy. See here, these are in the two plants in same species. Cross-pollination takes place between the two plants of the same species. This pollination is known as xenogamy. And second one, hybridization. See here, this is hybridization pollination. When cross pollination takes place between two plants of different species or different genera, it is called hybridization. This is cross pollination. Okay, students, are you understand? Pollination. Pollination is transfer of pollen grains anther to stigma of the same flower or different flower. That is pollination. Pollination is two types. First one self-pollination, second one cross-pollination. Self-pollination again divide two types, autogamy and zytonogamy. And cross-pollination also it is divided two types, xenogamy and hybridization. This is pollination. We'll go with next slide. Self-pollination, autogamy and zytonogamy, and cross-pollination. It is divided in two types, xenogamy and hybridization. We'll go with next slide. So see here, pollination advantages and disadvantages. So in this pollination occurs, some of the advantages are there. So first one advantage, it is a sure method of pollination in bisexual flowers where the sex organs mature at the same time. The male and female sex organs mature at the same time. And there is a least wastage of the pollen grains. And next one advantage, plants do not have to spread energy on special adaptations like color, nectar and fragrance. And one more, plants do not have to depend upon external agencies. And genetically pure line varieties can be obtained in pollination process. Once improved variety of crop or seed or fruit, it is obtained. Increase in the number of the same variety can be achieved only by self-pollination. These are advantages of the pollination process. And what are the disadvantages of pollination? So see here, it is not possible to introduce desirable characters in the progeny. And it is not possible to eliminate undesirable characters from the progeny. And continues the self-pollination and generation after generation results in the weak progeny. This is disadvantage of the pollination process. And one more, self-pollination produce comparatively less number of seeds, which are poor qualities and less viability of pollen grains. These are disadvantages of the pollen grains. So more than advantages in pollination process occur. And next one topic, agents of pollination. The pollination process occurs, some of the agents are needs. So these are agents biotic and abiotics. See, plants use two abiotic and biotic agents. The abiotic agents, these are water and wind. One more biotic agent, animals, to achieve the pollination process. But in majority of plants use biotic agents for pollination. Example, animals, insects. Only some of the small portion of the plants use abiotic agents. These are abiotic agents are water, wind. Next, pollen grains coming to contact with the stigma is chance factor in both wind and water pollination. So first one, agents of pollination, anemophily. So what is anemophily? Pollination, you know, pollination by wind. This is anemophily. It is more common amongst abiotic pollination. The wind pollination is also required that pollen grains are light and non-sticky in nature so that they can be transferred in wind. They often possess well exposed the stamen so that pollen is easily dispersed into wind. This is anemophily pollination. And second one, agents of pollination, hydrophily. The hydrophily pollination by the use of agent water. The hydrophily is quite rare to flowering plants and it is limited about the 30 genera, mostly in monocotyledons. These example, Vallisneria, one more example, Hydrilla and Jospira. But not all aquatic plants use water for pollination. We'll go with next slide. So in this hydrophily, pollination is divided into two types of hydrophily. First one epihydrophily and second one hypohydrophily. What is epihydrophily? In majority of aquatic plants such as water, example hyacinth and water lily, these flowers are emerged above the level of water and by pollinated by the insects or wind as the most of the land plants. The female flowers are reach the surface of the water. See here, the female flowers reach surface of the water by the long stalk, and the male flowers are released surface of the water. Some of them eventually reach the female flowers and the stigma. This is known as epihydrophily. And second one, hypohydrophily. The female flowers are remain submerged in the water and the pollen grains released inside the water. Pollen grains in many such species long and ribbon-like, and they are carried passively inside the water. Some of the rim reach the stigma to achieve the pollination. So this pollination is known as hypohydrophily. Example, Vallisneria, hyacinth, and water lily, and Jostira. And next one, agents of pollination, zoophily. So these are pollination by animals. In majority of flowering plants, use a range of animals as pollinating agents. For example, large animals and buffaloes and human beings also. And next one, agents of pollination, entomophily. Pollination occurred by insects. So this pollination, for example, bees, butterflies, and flies, beetles, wasps, and ants, and one more, moths. These are pollination occurred by the insects. This pollination is entomophily. And another next agents of pollination, that is ornithophily. These ornithophily pollination occurred by birds. For example, sunbirds and hummingbirds. And next one, chiropterophily. These agents of pollination occurred by the bats. And next one, agents of pollination, pterophily. This pollination occurred by the primates. And next, ophiophily. This pollination occurred by the reptiles and snakes, and example ginkgo lizards and garden lizards. Also have been in pollination occurs in some of the species. These are agents of pollination. Anemophily, hydrophily, epihydrophily, hypohydrophily, and zoophily. These pollination occurred by the animals. And entomophily, these pollination occurred by the insects. And ornithophily, these pollination occurred by the birds. And chiropterophily, pterophily, and ophiophily. These are all agents of pollination. In majority of insects, pollinated flowers are large, colorful, and fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered in the inflorescence to make the conches. These are agents of pollination. We will go with next slide. Next one topic, outbreeding devices. In majority of flowering plants to produce hermaphrodite flowers, and pollen grains likely to come in contact with the stigma of the same flower. So in flowering plants have developed many devices or adaptations or contrivances because of to discourage the self pollination, to encourage the cross pollination. Discourage the self pollination to encourage the cross pollination. In some of the species, pollen released and stigma is receptivity. It is not synchronized. This is known as dichogamy. That means dichogamy means pollen grains released, stigma is receptivity, are not synchronized. This condition is known as dichogamy. These dichogamy divide in two, protandry and protogyny. So what is protandry? Pollen is released before the stigma becomes receptive. This condition is known as protandry. And what is protogyny? Stigma becomes receptive much before the release of pollen grains. This condition is known as protogyny. Protandry example, sunflower. And protogyny example, datura and solanum. Protandry example, sunflower. Protogyny example, datura and solanum. In some of these species, anther and stigma are placed in different position so that pollen cannot come in contact with the stigma of same flower. This condition is known as hercogamy. Example, hibiscus. See here. One more outbreeding device, heterostyle. These heterostyle to prevent the inbreeding in the styles of the flowers in the same species are different heights. This condition is known as heterostyle. See here, this is long style. This is short style. So different heights in styles. This is short style. This is long style. These are male reproductive organs, anthers. This is female reproductive organs. So these device to prevent the self-pollination. This is also hetero-style device. Each flower has type on different plant. This is short style and long style. This is described of into the self-pollination. See here, these are male reproductive organs, pollen grains. These pollen grains transfer to the stigma of the flower. These pollen grains transfer to the pollen tube. And one more device, outbreeding to discourage the self-pollination, to encourage the cross-pollination, that is declining. This declining prevent the self-pollination is the production of unisexual flowers. This is known as declining. And next one device, self-sterility or self-incompatibility. That means to prevent breeding. So if both male and female flowers are the present on the same plant, such called monoecious plant, and male and female flowers are present on the different plants, that is each plant either male or female, that is dioecious plant. This condition prevents both autogamy and zytonogamy. That means the self-pollination is discouraged, the cross-pollination is encouraged. And these are 4 devices of the outbreeding devices. First one device, dichogamy. The pollens are released, stigma is receptive, are not synchronized. So dichogamy is 2 types, protandry and progyny. And second one device, hercogamy. The pollen cannot come to contact with the stigma of the same flower. This is hercogamy. And third one device, hetero. And third one device, hetero style. Flower have different types of heights of the styles and stamen. And fourth one device, decline. And fifth one, last one, self sterility. These are all discourage the self-pollination process and to encourage the cross-pollination. We'll go with next slide. See, protogyny. See here, stigma is receptive, but anthers are not shedding the pollen. This condition is protogyny. Example, bluebell. Stigma is ripens before the anthers. By the time of anthers, their pollens or stigma is no longer receptive to it. This condition is known as protogyny. And next one, protandry. See here, most of the flowers use this mechanism. Example, rose bay willow head. These stamens are ripened before the stigma is receptive to pollen. So the pollen is gone by the time of stigma is ready. This condition is protandry. Next one, pollen pistil interaction. This topic, pollination does not guarantee the transfer of right type of pollen, often the pollen of the wrong type. So the right type of pollen, it pistil has ability to recognize the pollen. So it is a right type of compatible type of the pollen pistil interaction. So wrong type, that is incompatible, it is a accept the pollen and promotes the post fertilization events. It leads to fertilization. So now we have discussed the pollen tube entry. The pollen tube after reaching the ovary enters the ovules through the micropylar region. This is progamy. See here, this is ovule, this is embryo sac. This embryo sac is haploid in condition, 8 nucleated and 7 celled. These are embryo sac. This female reproductive organ. This is pollen tube, enters into the ovule through the micropylar region. See here, this is micropylar region and then enters into embryo sac by destroying one of the synergy cell. This is known as progamy. Example, hibiscus. And second one, pollen tube entry, salazogamy. That means pollen tube enters into ovule through the salazal region. Example, casuarina. See here, this is salazal region. In this salazal region, it presents antipodal cells. And third one, pollen tube entry. See here, these are pollen tube. These pollen tube enter into the ovule through the integuments. These are integuments protect to the nucellus and embryo sac. So the pollen tube enters into the ovule through the integuments. This pollen tube entry is known as mesogamy. Example, cucurbits. Total 3 types of pollen tube entry. First one progamy, second one salazogamy, and third one mesogamy. So progamy, the pollen tube enter into the micropylar region And chalazogamy, the pollen tube enter into the ovule through the chalazal region. And mesogamy, the pollen tube enters into integuments. These are three types of pollen pistil interactions. So this embryo sac, it has 7-celled, 3 egg operators, 1 egg cell and 2 synergid cells. These synergid cells, it is presence of into the filiform apparatus in micropylar part of the synergids. These synergids, it is guides to entry of pollen tube. All these events from the pollen tube deposition of the stigma until the pollen tube enters the ovule are together referred as pollen pistil interaction. This is pollen pistil interaction. And next one topic, artificial hybridization. So do you know, students, artificial hybridization? See here, this is artificial hybridization. This artificial hybridization to help us improve the crop management program. In such crossing experiment, it is important to make sure that only desired pollen grains are used for the pollination process and the stigma is protected from the contamination from these unwanted pollens. This is emasculation and bagging techniques. So see here, first one, artificial hybridization, emasculation. So what is emasculation? See here, in female flower, in female parents bears the bisexual flowers. That means male flowers and female flowers present on the one flower, that is bisexual flower. Example, hibiscus, scientific name hibiscus rosa sinensis. So see here, in the female parent, that is bisexual flower, removal of anthers from the flower bud before anther descends using the pair of forceps is necessary. This step is referred as emasculation. So here, pollen grains are transferred. This is bisexual flower, male reproductive organ. This is pistil. This is male reproductive organ, stamen. This is female reproductive organs. So the male reproductive organ, the anthers are removed with help as forceps. This process is known as emasculation. So emasculated flowers have to be covered with a bag of a suitable size, generally made up of butter flower. See here, the anthers are removed from the flower bud condition. This is process emasculation. After emasculated flowers have to be covered with a bag for suitable size, generally made of butter flip. See here, this is butter paper. This to prevent the contamination of its stigma with unwanted pollen grains. This process is known as bagging. When the stigma of bagged flowers attain receptivity, mature pollen grains are collected from the anther of the male parents, are dusted on the stigma, and flowers are rebagged. These fruits are allowed from the develop. See here, bagging condition, bagging techniques. This artificial hybridization, emasculation, and bagging technique and rebagged. In the female parents produced unisexual flowers, there is no need for emasculation process. These female flowers got bagged before the flowers are open. When the stigma is become receptive, pollination is carried out using dissolved pollen grains for the flower rebag. This is artificial hybridization used for the two techniques, emasculation and bagging. And next one topic, double fertilization. See here, what is double fertilization? See here, this is pollen grain germinating on the stigma. See here, this is stigma. These are pollen grains germinating on the stigma. This is pollen tube. After entering the one of the synergids, pollen tube is burst and release the two male gametes in the cytoplasm of the synergids. See here, this is egg operator. These are two synergid cells, one egg cell. In the synergid cell, you have the special thickenings of these are filiform operators. So, entering of one of these synergids, pollen tube is burst, released to the two male gametes. Here, this is two male gametes in the cytoplasm of the synergy. So, one of the male gamete moves towards the egg cell, and the nucellus of the male cell fuse with the nucleus, thus completing syngamy or fertilization. This one is the first fertilization. And next is formation of the diploid cell, that is zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuse with them to produce the triploid primary endosperm nucleus. This is fertilization, the fusion of three haploid nuclei is termed as triple fusion. Since two fusions are syngamy and triple fusion, it is a embryo sac. This phenomenon is known as double fertilization. In this unique of flowering plants, in the central cell, after triple fusion become a primary endosperm cell. These are develops endosperm, while the zygote is develop into embryo. See here? Endosperm, this is embryo sac. So to form the primary endosperm nucleus, so these are endosperm develops well, zygote develop on the embryo. This is zygote, zygote is developed on embryo. The zygote gives rise to pro-embryo and subsequently to globular shape of the embryo and heart shaped embryo, and finally it is matured embryo. This is double fertilization process. Okay students, are you understand double fertilization, syngamy and double fertilization. And next one, post fertilization structures and events. So in this post fertilization, following the double fertilization events to form the endosperm and embryo development and mature of the ovules and seeds evolve into fruits. These are collectively termed as post fertilization structures and events. So first one, endosperm. See here, endosperm develop precedes embryo development. The primary endosperm cells, it is divides repeatedly to form the triploid endosperm tissue. The cells of the tissue filled with reserved food materials and used for the nutrition of the developing embryo. In the most common type of endosperm development, that is PN, primary endosperm nucleus, undergoes the successive nuclear divisions to give rise to free nucleus. This stage of endosperm development is called nuclear endosperms. So subsequently, it is a cell wall formation occurs. Endosperm become a cellular. So cellular endosperm and a nuclear endosperm. Endosperm may be either completely consumed by the developing of embryo before seed maturation. This is called non-endospermic. Example, coconuts. And endospermic. Maybe it is a persistence of mature seeds. That is endospermic. Example, pea and groundnut and beans. This is endosperm post fertilization structures and events. And next one, development of the embryo. So these embryo develops micropylar end of the embryo sacs where the zygote is situated. Most zygote divide only after certain amount of the endosperm is formed. These endosperm adaptations to provide the nutrition to the developing of embryo. So in this embryo subsequently to the globular and a heart shape and complete mature embryo. So the seeds of differently greatly in early stages of the embryo is development, that is called embryogeny. It is similar to both monocotyledons and dicotyledons. In the stage of embryogeny is dicotyledon embryo. It develops from the mature embryo. This is post-fertilization events and structures. And next one topic, dicotyledonous embryo. See here, this is dicot embryo. A typical dicotyledonous embryo, it consists of embryonal axis and two cotyledons. See here, two cotyledons. The cotyledons are fleshy and full of reserved food materials in dicots. The portion of embryonal axis above the level of cotyledon is called epicotyle, and which terminates into the flimule. See here, this is flimule or stem tip. First apical bud of the shoot. This is flimule. The cylindrical portion below the level of cotyledon is hypocotyle. That terminates into the lower end of the radicle. See here, this is radicle. Develop the root tip. The root tip is covered with a root cap. See here, this is root cap. Radicle and flummule. Radicle is a root part and flummule is a shoot part. This is dicotyledonous embryo. So embryos and next monocotyledon embryo. See here, this is monocotyledon embryo. Embryos of monocotyledon possess only one cotyledon. Example, grass family, the cotyledon is called scutellum. See here, this is scutellum that is situated towards the on the side of embryonal axis. At the lower end of the embryonal axis has radicle. See here, lower end of the embryonal axis, radicle, and root cap is enclosed. Undifferentiated sheath is called coleorhiza. The portion of embryonal axis above the level attachment of the scutellum is the epicotyle. Epicotyle. Epicotyle has a shoot apex. It is a leaf of primordia. Enclosed the hollow of foliar structure is coleoptile. See here. Few leaf primordia enclose the hollow foliar structure is called coleoptile. The outer covering of the endosperm separates the embryo. The proteinaceous layer is called alluron layer. This is monocotyledon embryo. Next one, structure of the dicotyledon seed. See here, this is dicotyledon seed. This is outermost covering of a seed is called seed coat. The seed coat has yet two layers. Outer layer is called testa and the inner is tegment. The helium is carried of the seed coat through the development of seeds attached to the fruits. The above helium small pore is called micropyle. Seed coat consists of embryonal axis, two cotyledons. See here, two cotyledons. This embryonic shoot, two cotyledons. These cotyledons after fleshy and fruit of the reserved food material. This is dicotyledon seed. And next one, monocotyledon seed. So see here, this is monocotyledon seed. Generally, monocotyledon seeds are endospermic, but some of the orchids are non-endospermic. See here, the outer covering endosperm separate the embryo by the proteinaceous layer is called alluron layer. See here, this is alluron layer. This is coleoptile and coleorhiza. This is radicle. This is flummule. Okay, students, this is monocotyledon seed. And next, last finally, apomixis. So what is apomixis? Production of seeds without fertilization is called apomixis. Example, in the apomixis, without fertilization, ostraceae and grasses. And next one, parthenocarpy. Already we have learned parthenocarpy. The fruit production without fertilization of ovary is known as parthenocarpy. Example, banana, watermelon, orange, grapes. And last finally, polyembryony. The formation of more than one embryo in a seed is called polyembryony. Poly means many. So one embryo in a seed more than. That is called polyembryony. embryony. Example, citrus and mango, and more than examples are there. Polyembryony. Okay, students, are you understand? I hope that all are understood this class. Today assignment. First one, describe the process of fertilization in angiosperms. And second question, write a brief account on agents of pollination. And third question, discuss the various types of pollen tube entry into the ovary with help of diagrams. Three types of pollen tube entries are there. First one, progamy, second one, salazogamy, and third one, mesogamy. Thank you, students. Thank you for living. Thank you, one and all. I would like to thank you, our secretary, sir, who are given this golden opportunity. And thank you, sir. And thank you, DSR, also for giving this opportunity. And I also thank our Sarala madam, who is the coordination for the botany and who was always there for me to appreciate my work as well as to motivate me to take up the class very smoothly. Thank you, ma'am. And thank you all children who are forwarded their work. I hope will continue the same. All the best for your IP. Thank you. Once again, thank you for all.
Thank you, ma'am.
Okay, sir.